Hi, everybody. Welcome to Micro Seminar for July 25th, 2019. We've been a little bit back and forth over the summer, but rest assured that Dave and Lizzie and I are working to get this back on track as a more regular thing. And today we are fortunate to have I believe, our very first speaker from industry, Geof Hannigan, who works at the Merck Exploratory Science Center. He, before that, did a postdoc with Pat Schloss at Michigan. Before that, the PhD at University of Pennsylvania, and before that, undergrad at the University of Minnesota. So he likes it cold, and he has quite a bit of microbiome chops from people that have both presented on the seminar in the past and others that you may know well. So without any further ado, I'm going to turn that over to Geof. Remember to send questions to us over Twitter through #USeminar. Go ahead, Geof.
Sounds good. All right. Thank you very much. I'm just going to hit the screen share button here. Start. Okay. And then make this bigger. Awesome. Are you able to see that?
Yep, looks good.
Okay, perfect. Cool. Yeah, so thanks so much for the opportunity to come talk at the seminar series. Yeah, this is really cool, and my first time presenting at an online seminar, so I'm excited. This is great. So yeah, I'm really excited to talk about some of the work we've been doing over at the Exploratory Science Center, looking at developing approaches to extracting functional information from the microbiome, excuse me, through deep learning approaches. And then I'm going to touch on some of our just kind of more like preliminary data around some of our microbiome vaccine work that I'm really excited about over here. So yeah, a little bit of where I'm at these days. So about 2 years ago, I left my postdoc over at the University of Michigan and came over to Boston, Cambridge, to the Exploratory Science Center. So this is us. We are part of Merck, the big pharmaceutical company. And we do a lot of really cool, like what we'd call early stage discovery, kind of like academic-ish, biotech-ish research. So we're a small group. We're about 45 scientists. We actually right now only occupy this top floor up here. And then we're also currently getting some space down here. So, you know, in October, we're actually holding a microbiome symposium in the building, and that's going to be hosted down there. And so, you know, you can see we're really embedded with a lot of pharmaceutical companies and a lot of biotech companies and a lot of really great academic institutions. So we're really in a cool place for a lot of biology, a lot of science, biotech, things like that. Excuse me. So again, you know, there's this paper that came out, an audience paper in Nature Drug Discovery with Daria Hazuda, who's our CSO over at the Exploratory Science Center. And so, you know, if you were more curious about like who we are, what we're doing, this kind of gives a little overview, but I just wanted to highlight, you know, that, you know, we really are trying to focus on these emerging areas of biological science with a very disease agnostic scope and trying to be nimble in that we follow the data and kind of go where we feel we need to go. And of course, because we're in industry, all of this is just very therapeutic minded and very translational. So really figuring out, you know, not just how can we do really cool biology, but how can we use that biology to really push towards new drug discovery and new therapeutics and that kind of thing. So we're organized into 4 groups over here. The names are kind of broad. So essentially we have a molecular discovery group, which is a very chemistry kind of metabolomic oriented group. They do a lot of lead generation. We have an investigational biology group that has a lot of you know, chemists, but more like biologists, immunologists, microbiologists. they do a lot of hypothesis generation, a lot of the various experiments on the molecular biology side. We have an experimental biology group, which does a lot of target and lead identification. Excuse me. And then on the bottom here, we have the group that I'm a part of, which is the system biology group, where we do computational biology, and we do a lot of like data integration and sequencing and that kind of stuff. And so this is just to say that overall we're a very diverse group with a lot of different backgrounds, which makes it a really exciting place to be. We've got a lot of different kinds of chemists, a lot of different kinds of biologists, a lot of different kinds of computational biologists. And so that kind of lets us get at some exciting problems. So enough about where we're doing research, but what actually are we doing? So I don't think I need to tell this audience too much about what the microbiome is for introduction, but essentially just to highlight that when we're talking about the microbiome, we're talking about these microbes, these bacteria, viruses, fungi, etc., that live in and on our body. And this is just kind of a cartoon to illustrate with some pie charts and stuff. But essentially, over the years, the microbiome has been associated with kind of myriad diseases, especially things like C. diff and colorectal cancer, and others. And ultimately, largely at the crux of those associations between the microbiome and health and disease in humans, a lot of it comes down to the microbiome and its interactions with the immune system, and that's something that we're really, really excited about. Especially because the microbiome is in close proximity to the immune system and has a lot of back and forth, and a lot of different interactions. And so just thinking about how we can leverage that understanding and how we can use that information to again do things like drug discovery and things like that. So all this is to say that we really see the microbiome as a field for immunological therapeutic opportunity, right? So one area that I'll touch on now, is things like the microbiome and cancer immunotherapy. So there's a lot of papers out there very recently looking at, the association between the microbiome and anti-cancer immunity, as well as things like anti-PD-1 efficacy, which is obviously something of interest to our group. And so that's a really interesting area for opportunity. On the other side, we also think about vaccine response to the microbiome. So again, it's no surprise given what company we're at, but just thinking about, okay, what are the associations between the microbiome and vaccine response and the immune system, and what kind of mechanistically what's going on, and how can we leverage that information to make better therapies, better medicines, and to ultimately help more people. And so we think a lot about translating the microbiome to therapy in different ways. So we think about questions like, can we reverse non-responders to something like vaccination or immunotherapy or other drug areas or therapeutic areas as well? Can we reverse that to make more people responsive to the therapy? We think about things like biomarkers. So does the microbiome provide orthogonal biomarkers of response that we could leverage therapeutically? This is out there. There's a lot of groups in kind of the FMT, fecal microbiota transplant space. So that's one way you could get at this. I think this is a really interesting area, thinking about how the microbiome is even metabolizing drugs that you're about to be prescribed, and how can we use that information, again, to help better inform delivery of medicines. And then, of course, kind of the big one is drug discovery. So especially thinking about things like antibody targets and small molecules and small molecule targets and that kind of thing. And that kind of segues, especially at the small molecule side, into the area that I'm going to be talking about fairly heavily is, thinking about how we can get that small molecule functional, actionable, whatever your buzzword is, understanding out of the microbiome. And so we really have focused recently on biosynthetic gene clusters in the microbiome. And so, in short, these biosynthetic gene clusters, or these BGCs, are these clusters of co-localized genes within a microbial genome. So it could be fungi, it could be bacteria. We're really focusing right now on bacteria, but essentially all these genes together encode the machinery needed to produce this final secondary metabolite, which is a small molecule. Often these are like natural products and can be thought of as like natural product drug candidates or for target ID and that kind of thing. So by understanding and looking for these clusters of genes, we can get a bit more of a functional kind of chemical understanding of what's going on in the microbiome, and then thinking about how we can leverage that again, in a translational sense. So at first blush, it seemed like an easy problem, right? I mean, yeah, okay, go find your BGCs, no big deal. But there's a lot of complexities in BGC identification that have been known about for a while. And machine learning is usually a way to get at this. And so, there's a problem in kind of accurately identifying BGCs despite these complex gene rearrangements. So these are all thiopeptide gene clusters identified in this paper. But as you can see, they're all coming from different bacteria. They all have different gene content and different numbers and gene arrangements and all sorts of things. So it makes it analytically a difficult problem to solve in identifying those. Because they can be fairly diverse, there's also complexities in our capabilities to identify novel BGC biological signatures of our data, and just kind of going beyond what's known. When we were thinking about our machine learning, kind of deep learning approaches, we wanted to not just think about finding that gene sequence or that part of the genome that's a BGC, but we also wanted to think about, can we use things like BGC genomic context and other genetic elements to more accurately identify these BGCs as well as understand the biology. And so we saw an opportunity here in existing machine learning approaches. Maybe requiring improved accuracy is probably a little too strong, but essentially, there's this opportunity for improvement in kind of the existing algorithms and what we can do to offer the field and to improve our understanding. And so, we had this problem, and we did what oops, I lost my light in here. I don't even know if my video's on, so it might not matter, but there we go. There we go. But essentially, we did what any good scientist does, right? Which is you go onto Google and you ask, Google, how do I identify BGCs? But it's interesting when you go into Google and you start typing that out, you'll notice that it'll start trying to guess what's coming next, right? So there's no one on the other side that's doing this, right? It's just an algorithm that Google has kind of behind the scenes that's able to, quote unquote, understand what you're typing even before you've completed the word, and guess what you're likely to want to type next, and to guess like the sentiment, what it means, what the topic is, what its association to other words are. Even on this side, you could even like speak into it. And all that really gets into these deep learning approaches to, again, quote unquote, understand language at an algorithmic level, especially by incorporating context of words with other words within sentences and different levels of corpus of data. And so we thought maybe we can try treating genomes like language to better identify BGCs using these kind of natural language processing approaches. So again, on the English side, you can have a sentence, and algorithmically, you can go through and you can identify what parts of speech these different words are and how they relate to each other and where their pointers are. We wanted to take this kind of algorithmic approach and I apply it to kind of the genomic language. So again, trying to say, okay, instead of words, how are the genes related and what do they mean and what parts of the genome are they and that kind of thing. And then especially apply that at the biosynthetic gene cluster level. And so we set up a really fun collaboration internally. So over here on this side of the world, we had our Cambridge and Boston sites of the company. Our company is a global company, and so we really struck up a cool collaboration with our Prague colleagues over in the Czech Republic, who have a lot of expertise in kind of the natural language processing, machine learning kinds of approaches. And so as we started figuring out how we were going to tackle this problem, we kind of broke it down into, do we want to think about things at the genetic, like gene level? Do we want to think about it at the PFAM level, nucleotide level? Where do we want to go with this? So, we decided to go with the PFAM level. What we did is we built out this program we very, cleverly call PFAM-TVEC, which is based on Google's famous algorithm Word2Vec. But essentially, it's this embedding technique where we can take this corpus of data and we can actually build out an approach to essentially define these protein families instead of by their sequences, by their context within groups of other PFAM protein family domains. So you're defining them by where they are in the genome more than what they actually are at the sequence level. So that was a really, I think, helpful way to do this, and it let us get some really good, accuracy as far as classifying these protein family domains based on kind of their known functionality. Because a lot of these have been annotated and that kind of thing. So that was a pretty cool approach to get started with. The other, problem we had was, okay, we can go to like the MyBig database and we can get BGCs of known classes and that have been characterized. But where do we get the negative data? Because we want to try to trick this algorithm, right? And we want to tell it what's not a BGC just as much as what is a BGC. And so what we did is we built out our own negative training data set. We started by just pulling out sections of genomes randomly that were within a similar distribution of length as BGCs, but that was way too easy for algorithms to figure out. So what we did is we took, here with my super fancy cartoon, we took BGCs, so for example here in blue, where you have some genes in orange within the BGC and you have some genes outside the BGC within the genome. What we did is we took a given BGC, we popped out the genes, only leaving the blank scaffold. And then we randomly took genes from other parts of the genome that were about the same size as the original genes, and we popped those back into the BGC. So in the end, what you have is this synthetic BGC that we wouldn't expect to actually have any biosynthetic gene cluster activity because it's just a bunch of random genes thrown together from throughout the genes, throughout that same genome. So essentially, we have all of these null BGCs that we can use to train our algorithm and try to trick it to get better performance. And so we took the PFAM-TVEC algorithm and we took our negative training data set and we paired that up with the positive training data set, which came both from some previous publications as well as the MyBig database. And we built out this deep learning, what we, again, very cleverly call DeepBGC algorithm. So at a very high level, what you do is you start out with input as a raw sequence. So this could be a bacterial genome that you've sequenced out. It could be a contig from your metagenomic data set. You go through gene prediction on that genome. You break that down into protein family domains that are predicted. And again, you can identify those by PFAM-TVEC. We can go through our long short-term memory neural net, which I'll talk about in a second, to essentially get these BGC scores for whether or not we think these different domains are part of the BGC. We have a gene level summary score that incorporates where the gene positions are. And then together you get, this kind of candidate BGC selection. It goes through some optional post-processing that we wrote in there, but ultimately it'll say, okay, yeah, this is what I think a BGC is. And so again, kind of this middle part is based on this long short-term memory network. So you have your sequence of protein family domains. We go through the PFAM to VEC embedding process, and we have some vectors associated with whether this is associated with a gene start or a gene end position. This goes through our trained long short-term memory network, apply the sigmoid function at the end, and then we get this BGC prediction score that we can utilize to see whether or not these genes are together part of BGC or not. And there's kind of other filters there as far as how many genes are part of the cluster and things like that. But as far as performance goes, one of the problems we found, and I think most people found in the field using, we used this algorithm cluster finder, which is awesome. And we chose it because it's one of the leading algorithms in the field. But it can suffer from kind of sputtering along and having a fairly high false positive rate, right? So you'll be able to get BGCs, but you'll get things that are showing up that aren't really BGCs. And so here we have a manually annotated genomic sequence. We know where the true BGCs are, which are shown in gray. And then in the blue, we have that cluster finder algorithm prediction. So again, and as I said, you have kind of these false positives like over here where we know there's no BGCs over here, but it's showing up. And then in the red, we have our DeepBGC approach. And so overall, just highlighting that this is doing a better job at more accurately identifying the BGCs. And so we wanted to look at the performance in other ways as well. So what we did is we built out these ROC curves using training and testing data sets, and just really, briefly to highlight what that means. What we did is we took, our corpus of biosynthetic gene cluster sequences, which you can see here of various different colors. And then we have our non-BGCs that I talked about before, which are kind of these colorless regions. We then mix them together and split them up into a training and a testing set. And then we trained the model by providing, answers with the data, right? So we'd say, okay, machines, green is a BGC and red is a BGC, like colors are BGCs and gray is not. And then what we would do is we would test that algorithm. So we'd say, okay, now without looking at the answers, here's a purple one. What is that? If it returns BGC, then great. That's check mark. Awesome. You got it right. If we give it a gray one and it says BGC, then no, sorry, that's a wrong answer. And we know that you're having trouble accurately identifying BGCs after your training. And so we can use that information to get false positive rates and true positive rates from the testing set. Looks like it maybe got a little fuzzy there, but essentially what we have is this ROC curve where we're hoping to get these curves up into the corner where we have the 0% false positive rate and 100% true positive rate. Again, the cluster finder algorithm, or what I call here old method, is performing at a lower level. So this area under the curve, or this quantification of quote unquote accuracy, is much lower, so about 0.84, 0.87. And then our algorithm in red is performing much higher, up at about 0.92. You'll see that there's both a solid and a dotted line. So what we have here is the solid line being kind of the out-of-the-box implementation of that existing algorithm. And then what we did is we re-implemented the algorithm, based on HMM, and we retrained it on the exact same training data that our DeepBGC had, because we wanted to, right away kind of address the question of, well, hold on, maybe your method is just doing better because you trained it on newer data or different data. So we did that by retraining everything on the same data sets, trying to give a level playing field and make sure that everything's good. So we're also really curious about how well this type of deep learning contextual approach could identify novel biosynthetic gene cluster information, or novel BGCs, and then as a result, potentially like novel chemical matter. So again, we took our collection of BGCs and non-BGCs. What we did is we mixed those together, but we trained the model on all of the BGC classes except for one, and we withheld one of the classes. So as far as this algorithm is concerned, there's no such thing as like a RIP or something. That class is totally unknown. And so we go through and we train the model, and then we ask how well can you identify that class that we withheld. So by training on other types of BGCs, can you identify RIPs? And so we were really excited by this result. So we have here just illustrating the 6 big classes of BGCs that overall, looking at these rock curves, the DeepBGC in red, we were able to do a much better job across the board as far as identifying novel BGCs using this leave class out validation approach. So especially here in RIPs, by training on things that aren't RIPs, we're able to do much better as far as that identification goes. So we were really excited about that as far as that kind of novel identification approach. And this also gets at some of our concerns around, okay, if you're training on kind of the workhorse bacteria, and you're going to have a biased training set because it's going to be all the things that were used in the lab and all that. And so, by trying to make sure we're open to information that's novel and that we didn't have at time of training, just kind of hoping to address some of that. And so, when we went through and looked at applying DeepBGC to RefSeq genomes, we found that we're able to detect BGCs from a variety of bacterial taxa. So that, of course, there's a lot of these others which are miscellaneous, but you have a lot of like Microbiome and Bacillus and et cetera, et cetera. When we look here, so this is everything we identified as DeepBGC. On the right here, we have only the BGCs that we could identify with DeepBGC and that were missed by the anti-smash kind of cluster finder types of approaches. So you can just see that we're able to uniquely identify a lot of Mycobacterium BGCs, but also a lot of BGCs from other bacterial taxa, which was interesting. And so ultimately, what you want to do is use this to potentially identify novel biosynthetic gene clusters. So again, what we did is we went through and applied this to reference genomes that were just available, in NCBI. And over here, you can see this Tisney plot, this ordination of various BGCs. My light's out again, but whatever. So you can see, this ordination of BGCs. We looked through with especially one of our chemical collaborators, who's really an expert in this biosynthetic, natural product chemistry space, and identified, in this case, one example, BGC, although there's others, and I'll put a link to the preprint down here so you can check out others if you're interested. But, we found this BGC using DeepBGC. We had built a prediction algorithm that predicts activity. I didn't really talk about it here. That's also in the preprint. It's not a great algorithm, honestly, just because of a lack of like training data and things like that. It does okay, but not stellar. And then again, we did some manual confirmation. Is this being predicted for antibacterial activity? And yes, the experts we work with figure, yeah, this is probably a new antibacterial BGC that we identified. And here's just kind of a cartoon image of what it looks like. So yeah, so that's a really cool application to even looking at known genomes from the NCBI and being able to identify new material. So again, as I mentioned, we have this out there for people to check out. So we have a DeepBGC GitHub repository. So if you want to check out the source code, you can totally do that. You can go here, and I think you can just like Google DeepBGC, it'll probably show up. So that's openly available to people. You can also install it like through Python and that kind of thing. So we hope easy to load in and use. And from what I've heard from some folks, it seems like they're getting some cool results, external to our company even, which is neat. We have this on the bioRxiv. As you can see, it's been on there for a bit now. We actually are pretty much done publishing this. So, if you keep an eye on Twitter, I'll try to put an announcement out there for when the final publication is out there, but you can get the sneak preview here on the bioRxiv. So just to kind of wrap this part up, we see leveraging this kind of deep learning, even though I know that's kind of a buzzword, but leveraging deep learning in microbiome genomics as being an advantageous approach. So, we found that we're able to reduce false positive BGC identification using this type of approach. We can extract BGCs from genomes and even metagenomes more accurately. We feel like we can identify novel BGCs a little bit better. Excuse me. And then, ultimately, what we're thinking about is kind of leveraging this machine learning at the microbiome level. So thinking about things like immune modulation, target identification, and that kind of thing. And so, we see a lot of opportunity here as well for continuing to build on this. So as we get more data, we can scale this up and train the model to be better. So that's great. We built a thing. Hopefully it's helpful, but where should we actually be applying this? Like where does kind of the rubber hit the road? So, as I said, we're interested in kind of this immunomodulatory host interaction space. Excuse me. So, when you look at the literature, you can see this, I guess, what we consider emerging published evidence for both direct and indirect impacts of, for example, antibiotics on host immunity, and presumably that has a microbiome component to it, right? And so, I think we've all seen these types of papers where you have the antibiotics impacts on things like pathogen outcome. This paper is especially relevant to the vaccine world, but antibiotics impact on vaccine outcome and lower antibody responses associated with antibiotics exposure, and then these kind of direct effects of antibiotics on immune cells as well. So we've been curious in trying to measure the association between the vaccine response and the microbiome, and thinking about how we can develop like animal models for this and really establish kind of some foundational understanding. And so here I'm just going to highlight a couple kind of cool experiments that we had run. So the first was just kind of starting out and looking at measuring the impact of different antibiotics on vaccine response, internally in our company, right? So something I'm sure a lot of you have heard of, or if you're working in the industry, you might be aware that, a lot of the rigor of starting things out is around, does this work in our hands? Are the publications reproducible? Can we actually do it? So this was kind of in that vein, but also, we feel like we're adding some information here, which I'll talk about in a second. So essentially what we did is we started our antibiotic treatment at what we're calling this day -8, which is about a week before vaccination. So you're on antibiotics, excuse me. We allow, for about a week, we allow an antibiotic washout period. And then what we did is we took a stool sample at the time of this initial vaccination. And then we did a booster vaccination a couple weeks later. And then finally we did this vaccine antibody titer, to see how well the vaccine responded. And this was all done with a OVA experimental vaccine. So it's a model vaccine. And so, as you would expect, after we did some 16S rRNA gene sequencing, on these stool samples after antibiotic treatments, looking at kind of a slew of antibiotics, we had a variety that we used here. We found that the different antibiotic treatments yielded very distinct microbial communities, which again I think is expected, but it's nice to see, right? So if you're treating, with I don't know, like vancomycin, you're kind of over here. With your Bacterial, you're over here. You got your tobramycin, your neomycin. So it's kind of just nice to see that you're getting these what we consider like distinct microbial communities after different antibiotic treatments, and that's important then when we think about distinct vaccine responses. So here what we have is vaccine titers, again IgG against the OVA vaccine, and just ordered by mean response. But here you have your water control, right? So these are what the vaccine responses in the mice would look like if you didn't treat with antibiotics, if you just treated with water. And then as you give different types of antibiotic treatments, you see these variable responses, right? And interestingly, vancomycin, which other groups have identified as well, really produced the greatest dampening of that IgG response in the mice. So this was really cool. It was cool to see kind of that gradient, right? That with the modulation of these microbiomes in very distinct ways using different antibiotics, we're able to see more or less kind of these distinct communities. And I won't talk too much about like correlations and stuff you could do, but you can do those kinds of things here as well. And so, okay, I give different kinds of antibiotics to the mice, and I get different dampening of the vaccine titers, but can I kind of do the opposite? So, instead of thinking about the microbiome getting depleted in different ways and then trying to associate that with microbiome response, can I deplete the microbiome in the same way? So in this case, antibiotics means vancomycin, with the same length of treatment, but can I vary the what we call washout, or you could say like recovery time? Could I vary that before the time of vaccination? And is there an association between microbiome recovery and vaccine titer? And then what we did here in this 3-in-1, we also had an arm where we did a 3-day treatment of antibiotics instead of a second 7-day. And then we'd also do like a 1-day treatment of antibiotics. And again, all vancomycin here. Then you get your vaccination and you go on your vaccination regimen and you get your titers at the end. So this, again, an encouraging result that the microbiome, in these guts tended to recover, towards the baseline state over time and kind of follow these predictable dynamics. And so, what we're looking at here is an OTU dissimilarity measurement using the Bragg-Curtis metric, but looking at mean dissimilarity of those stool samples compared to the water control. And so here you can see everyone's kind of in the same place and they're all fairly similar. I don't have the water one here, but all, at least to the same degree similar to the water controls. So the washout day is how long the recovery was. So the green was the earliest treatment of antibiotics, and I think you could have guessed that by looking at this. But essentially you can see this increase in dissimilarity, right? So it's getting much further away from that, quote unquote healthy water state. And then when you stop the antibiotics and allow it to recover, it goes back down towards the original state. And then the same with the others. And again, you can guess at what time the antibiotics were delivered. And then interestingly, never actually getting back to the total level of similarity as it originally started, which I think, is probably expected in a lot of ways. But kind of getting down to a dissimilarity of about 0.75 instead of 0.5. And so also just to focus here at day 0 again, where vaccination is, when you allow for different recovery times, you have this gradient of microbial communities that is more or less similar to what it would have been at a healthy state, right? And that's important again when we think about looking at titers. So here we again have the IgG over response at the end of the study. So what we're looking at is titers to the vaccine, with the blue line being the mean. And again, is pretty cool, right? That here's your water control baseline. And if I jump over to the right side over here, with the longest washout period or the longest recovery time of 21 days, you can see that the titers are pretty high. You know, so getting up there to around where the water is. And then when you shorten the recovery time, you see an overall reduction in titer. And then again, when you shorten the recovery time after antibiotics, and therefore having a more, I guess, disturbed community, you have even lower titers. And then when you do the week of antibiotics with just the 1-day recovery, you see the lowest, right? So again, not only can we use different antibiotics to differentially, I guess, knock out the microbiome and associate that with vaccine titers, but you can also then modulate the recovery time and get increased titers with microbiome recovery. Great, and so to kind of wrap this part up, you know, ultimately, you know, we're working on understanding and leveraging this link between microbiome and vaccine response. So what I showed you here were some of the more kind of preliminary establishing types of results, but ultimately, you know, the goal here is to think more, you know, functionally about this and using a variety of techniques, but especially thinking about using our DeepBGC algorithm in this space, you know, thinking about what types of BGCs and products are associated with the response side of the spectrum, and then how does that look in the non-response side of the spectrum, and then how can we actually, you know, act on that information? So what can I go back to the bench and do as far as experiments go to establish that causal relationship, and then ultimately, you know, move that through kind of the translational approach of identifying, you know, what targets are there, or small molecule drug candidates and that kind of thing. So again, overall, just kind of highlighting that we've consistently observed this link, and, you know, in this data and as well as data that I'm not showing. You know, we're, as I said, interested in understanding what microbiome factors are correlated or otherwise associated with vaccine response. And then, yeah, again, can we kind of reduce that relationship down to practice? You know, can we actually translate that into medicine and targets and things like that to improve, you know, vaccine response, which is still a big problem in a lot of ways. So with that, I'll wrap it up, and we can take some questions. But, you know, just to acknowledge the people that have worked on this, because this has just been a huge effort, you know, across the company. And so here at the Cambridge Exploratory Science Center, we have a variety of folks who worked on this team, as well as this DeepBGC is a group in Boston, as well as cheminformatics. We've worked with some business development people, a lot of folks, as I mentioned, over in the Czech Republic and Prague. And I like to highlight, you know, we have this cool group internally. It's kind of a smaller kind of grassroots group, but they call themselves Prep Talks, and they kind of help you go through your slides and give you like pointers on presentation and things like that. So I thought that was a cool resource. So I try to highlight them and just thanking them for hopefully helping me be more, you know, more effective as a speaker to audiences. And then just kind of continuing those acknowledgements. So, you know, this antibiotic and vaccine team contribution, we have folks again from the Exploratory Science Center in Cambridge, as well as folks down in Pennsylvania, as part of this infectious disease and vaccine group. There's a lot of other additional contributors and groups that we've worked with, which has been really wonderful. So yeah, with that, I can take any questions. Do I usually leave the slides up, or should I stop sharing? I guess
go ahead and drop out of there,
yeah.
so we can see your mug and
Yeah.
ask you questions face to face.
Okay, sounds good. All right, perfect. There we go. Cool.
Great. Well, thank you very much, Geof. That was really neat to see the development of the tool and then some application. Does anybody inside the Hangout have any questions? I think we have a couple people with us in here. I don't have any questions from Twitter, but if you are watching and you want to send something through that way, just make sure you send your tweet to #microseminar or add us at @microseminar on Twitter. And while we're waiting for that, if you are in the room and want to ask a question, just unmute yourself and interrupt. But I was thinking, since you are the first speaker from industry and you've definitely walked through the halls of academia on the way in, it would be, I'm personally curious how you like working in industry, and obviously this is being recorded and
yeah, right.
So
yeah, whatever I say can and will be used against me.
Yeah. So everyone's going to hear it, but maybe some reflections on what that transition's like for those who are potentially reviewing this in the future and are considering the same type of a move.
Absolutely, yeah. I love it. I love where I'm at. It's been a lot of fun. I feel like I get to do a lot of cool science, and then also I get to learn a lot, which is awesome. I think one thing I was told before I went into industry, but I think it's rung true, is this idea that at the university level, when you look at different universities and how things are run, that seems to be more of shades of gray as far as how things are done. And that's to say I could go from UPenn over to Michigan, and I'd probably have a pretty decent idea of how the department would be structured and what the levels are called, associate professor and things like that. Whereas industry can be very black and white. Companies are running themselves in sometimes very different ways. And so all that is to just say that, I could offer my 2 cents, but it could absolutely be very different as soon as you get into a different space. No, I mean, I've loved it. I think one of the things I've noticed being in a big company is you do get to do very collaborative work, which is wonderful. I'd say most things are done at a scale larger than what I was used to in academia.
Yeah.
So that's really cool. And with that comes, additional pressures against, things like managing a project and time management and things like that. Another thing that we think a lot about is, when I was at the university, you could kind of say, what I am doing could be used for this, right? It's going to help the world in this way, and it's going to be a drug, which is awesome. But I think, being at a company, there's a little bit more pressure to, not just say that, but to try talking to the people, right? Because at the company, you have that range of people all the way from discovering the thing to making the thing, to selling the thing, to marketing the thing. So there's just a little bit more exposure to okay, I could say that I'm going to make this a drug or that it's going to help out. But because the resources are there, there's just kind of the opportunity to actually go ask someone further down the line to be is this actually what I think it is? Could you make this, would it actually be a medicine to someone, or is it not able to be scaled up or something like that?
Right. Or it's not feasible in the market the way it is.
Yeah, exactly. I mean, if it's I think cynically, I think sometimes people think it's like all business questions, which I think it really is about, people and trying to get a medicine out there. But yeah, if it's just not feasible, if there's something already out there and you're not really doing better, then that's not a need for people.
Yeah.
Yeah, I don't know, just some 2 cents, I guess.
For sure. Yeah. No. Cool.
Yeah.
I actually do have a science question.
Absolutely.
Just to keep the ball rolling here for a couple more minutes, but and you probably, this might be in your paper, but I was wondering if you could tell me about the range of taxa that you've tested your cluster finder on.
Yeah, so we tested it on the RefSeq database of reference bacterial genomes. So as diverse as that is, but that being said, there's not like huge amounts of like really like manually curated, training data and testing data and things like that. So, there absolutely are biases in the databases and things like that. So it seems to work pretty well across different taxa, and we hope that kind of that novel discovery leaf class out validation thing I showed also helps contribute to that. But it's something we think about, absolutely. And then how can we get more data to figure out how to address that? Yeah.
Okay. Yeah. I mean, we're definitely going to be. I'm going to have us start using that on all the genomes that we produce, just as a secondary check. Yeah.
That'd be great. Let me know how it works, and if you have any issues too, let us know. We can.
Yeah. We're good test. We're a good test group for installing software because we're not super computationally savvy. So if there's a way to break it, we break it.
Awesome. That's fantastic.
Cool. Well, if there's, let's see, I don't think there's any more on Twitter. Let's see. No, I don't think so.
Well, if people want to ever like reach out, because I know it's recorded, I'm on Twitter like at iProphage, or you can Google my name or whatever. So always happy to chat if people want to. If it's like 2 years from now you see this video.
Right. No, that's great. We usually
feel free to reach out.
We usually ask people how they can get someone can get in touch with you if they do check this out a little bit later. So that's great.
Yeah.
Okay. Well, thank you very much, Geof, for your time and your great presentation and contribution to the series. And best of luck. And I'm sure we'll see you in the near future.
Awesome. Thanks so much. This is great.
Take care.
Talk to you later.